So welcome everyone this evening. My name is Claire Thomas and I've been here since last a week about a year ago, July. And I really came here because I was so impressed with the board and obviously meeting Mary for the first time, who's the first ever executive to make me a cup of tea. So that sold it to me. So I just want to thank you both for your very, very kind introduction this evening. I'd like to talk briefly on an area which I'm extremely passionate about, which is resistance to resistance. So it's basically gram-negative resistance in our bacteria that we're all colonised with. And I'm going to whiz through in about 20 minutes, so forgive me if I speed over things a bit. So essentially, you've all seen these rather scary headlines the last few years. How our hospitals are losing the war on bugs, etc., scary stuff. But I really want to say that we're actually doing very, very well in our hospital. So before I panic you all, I'm going to talk about all the scary stuff, but actually here we're doing very well. So please don't worry. So I want to talk about why we're worried about resistance organisms now, and I'm going to talk you through some very quick microbiology. Any microbiologists present? And also about, well, do see my colleague there, do we? And also a brief outline on antibiotics and their history, and what are we doing about the rise in antibiotic resistance, particularly in our gram-negative organisms, and what can we and what can you do as patients in the community. So the big thinkers in 2016 declared a state of emergency around antibiotic resistance. The WHO put a lot of bulletins out. The Public Health Laboratory here at PHE put out some edicts, etc. And we've been worried about this really for the last 10, 15 years. And the UN had very high-level meetings in 2016 as well, where they tried to look at the causes. Why are we here now? Why are we running out of our antibiotics? Why are we looking at a possible post-antibiotic era? And essentially our real problem is we are fundamentally running out of treatment options for infections caused by previous sensitive bacteria. And to understand bacterial resistance, we have to understand bacteria. So if you forgive me, I'm just going to do some basic microbiology. Karen, please. We're going to do a whistle-stop update on microbiology. So our laboratory is amazing. It's one of the nicest labs I've worked in my life, and I've worked in a few. It essentially does some quite old-fashioned things, which is still fantastic, but we do a lot of molecular work, clever polymerase chain reaction serology. But essentially, the fundamental truth here is we're doing everyday gram stains on the bacteria that are coming out of people's blood samples. So you take a blood sample if I have a fever, and it will go into a special bottle, go on one of our culture machines, and it will ping positive by dint of a quick photometric assay, and we'll do a gram stain. It's just a simple 30-second stain. It will tell us what's in there. Is it gram positive or is it gram negative? And it's the gram negatives I'm going to talk about today, the gram negatives that are in all of our guts, the E. coli's and the Klebsiellas particularly. And we know now, we used to think it was 10 to 1 bacteria to human cells, it looks to be 3 to 1 bacteria to human cells, and most of the time we coexist very happily, colonisation, not infection. And they know now from some quite clever metagenomics from the Human Genome Project that there are various niches in our bodies. So my skin now, if you take a swab of my skin, there are various bacteria, but they're causing me no problems. They're very low pathogenic bacteria. If I then have something like diabetes and I've got a cut, one of the bacteria called a Staphylococcus aureus or a group A strep may zip in and cause me trouble. But most of the time, the bacteria behaving cells, it's a mutualistic, synergistic relationship. The main resistant microbes that we're worried about now in 2016 are the gram-negatives. So essentially, we have very few antibiotics left for our gram-negative infections. The gram-positives, MRSA, VRE, big scary stuff, we've got antibiotics for those. We can still treat them. In many parts of the world where I've been working, like Singapore, we have antibiotics, we have bacteria, E. coli, Klebsiellas in our gut, which there's no antibiotic for. And then we're looking at a post-antibiotic era. Often all we can do is perhaps get the surgeon on the line and debride or whatever. We're using antibiotics, very, very toxic, old-fashioned antibiotics, but it's an uphill battle. In the UK at the moment, we have issues in certain of our hospitals, not here, with these very resistant bugs, but this is something we've got to be aware of, got to keep an eye on. So the gut microbiome, I just wanted to focus on this for a second. The gut is far more complex than we originally thought. It's only 6 to 7 metres long. I thought it was longer. And essentially the whole of the surface area is huge. So it's about half the size of badminton court. What's the ideal composition for your gut? Well, people are still trying to work this out. If you're interested, there's a very interesting piece of work by Professor Tim Spector, who works at King's College London. So he's got some ideas coming out of twin studies. But it seems that we have over 1,000 different bacterial species in our gut, and essentially some 10 trillion bacterial cells. It's a real big thing to get your head around. But it's really a balance of our old favourites, the Escherichia coli, a few Klebsiella, the gram-positives enterococci, and a bit of yeast with gram-negatives all mixed in. And the E. coli, this is one of our main concerns at the moment. It normally is quite happy in our large gut. It does well from us. It uses nutrients that we absorb every day, and it makes vitamin K and B back, so it gives us back something. And fundamentally, it always protects us against other bacteria, the pathogens like Salmonella and Shigella. So this so-called mutualistic relationship is something to hold in mind. But if it gets somewhere else, like our urinary tract or our biliary tree, or if we have a lining infection or something, we get in trouble. So this is our main cause of community gram-negative sepsis coming through the door. So this is something I wanted to have a quick look at. And a quick bit of history, to look forward, we must look back. Penicillin's been only around since the 1940s. It was first discovered by Fleming as a so-called mold juice. We think that he may have left, he went away for a few weeks. He left a plate of Staph aureus up, and he noted that there was an area of killing around this mold, and it was that area that they eventually purified penicillin from, which is a beta-lactam antibiotic. It's one of the building blocks of all our antibiotics. And the golden age followed. The chemists got hold of penicillin. We got chloramphenicol. We got macrolides. And the thing about the beta-lactam antibiotics, the basic penicillin, the chemists started adding lots of special groups onto that. So we ended up with this workhorse antibiotic, which we used a lot in the 80s, called cephalosporins. But just hold on to this picture, because what we didn't realise, and I think what the original microbiologists realised, is that resistance predates antibiotics. So it was discovered that there were E. coli resistant to penicillin even before they used penicillin. And recent studies have shown that if you take samples from all of us, even if we haven't been exposed to antibiotics, there are antibiotic resistant genes already present. So the bacteria are cunning, they're clever, they're ahead of the game. The main mechanisms of resistance, I don't want to bore you with this, the main thing is there are all sorts of mechanisms, but the bacteria replicate every 2-3 hours, and they can change genetic information. And you'll see a picture of the Death Star for Star Wars fans down there. That's representing a bit of plasmidic. It's a small circular piece of DNA that these bacteria can carry many, many, many copies, but on that bit of circular DNA are many, many resistance genes. It's not just for one antibiotic like penicillin, it's for other types of antibiotics as well. So these are really nasty. And the bacteria share genetic material. So this is bacterial conjugation, and many, many copies of these different plasmids carrying all these resistant genes are being exchanged. And what we found is that the different bacteria are sharing genes. So the E. coli has picked up bacteria from soil organisms. It's picked up bacteria from Klebsiella, and they're exchanging them all the time. They're not exclusive. So penicillin was used as a universal panacea until the 1960s in Japan, when they realised they again were seeing the rise of penicillin resistance in the E. coli. And then unfortunately the cephalosporins, for those chemists amongst you, it's just penicillin with a few R groups added on. Unfortunately now we've got resistance to those. So these are workhorse antibiotics. So the beta-lactamase mechanism we've got, and that's called an extended beta-lactamase, so they're called ESBLs. So I'm sorry about geeks. A lot of geeks speak here, okay? I'm skimming over this because it's a personal area. But we're basically seeing resistance to our workhorse antibiotics. And the public health labs realised that the E. coli bacteremia being sent through to them were becoming more and more resistant in 2000, and in the Klebsiellas. But the real worry was we were seeing resistance in the hospital setting, fine in Klebsiellas, but we're seeing resistance in the E. coli in the community. So something's happening in the community. This was people with no contact with hospital setting. So it seems like it's a continuum. There are reservoirs for these resistant bugs in the community, and in the hospital it's a continuum. But what did it mean for our patients? You know, this is all lab stuff, theoretical stuff, but we know from work by Patterson in 2002 and 2003, patients don't do very well if they have these resistant bugs. And one of the fundamental problems was until 2004, the laboratories were not able to look for them terribly well. That has now changed. And I can assure you that our micro lab is picking these up as quickly as they arise. Our problem was, yes, we can rely on these end-of-the-line antibiotics, so this is fine, okay? We still had the so-called carbapenems, which were a form of penicillin, and this very old-fashioned drug called colistin, nasty, toxic drug, but it was still, we could still use it in treatment, and I used it quite a lot in Singapore. The problem now is the use in the poultry industry seems to be that we've lost even our end-of-the-line antibiotics, colistin. So are we at the end of the line? Are we looking at the post-antibiotic era? Well, I'd like to say we're very close to the end of the line. What can we do and how can we resist the resistance? And why are we in trouble now? Well, the background to this really is we're overusing. There's Darwinian selection. The bugs already knew how to be resistant. So they can change genetic material. Overuse in the food industry, the animal industry. DEFRA's looking at this at the moment. We all global travel. I mean, I was very, very privileged to be able to work in different countries around the world. Heaven knows what my own gut bacteria are doing, but we've not looked. Resistant organisms have not been detected in the lab in the 2000s, but that's now been picked up. There's a huge underemphasis. I remember doing my big exams for medicine in 1998 and not being expected to wash my hands between patients. I mean, that's just crazy. Now, it's automatic. There's no way I would go and introduce myself to a patient unless I had gelled my hand and I'd been bare below the elbows, taken my watch off, and I'd also gel my hands coming back out. You know, so this is fundamental. It's about changing behaviour. And there are fake medicines in the third developing world. We know about this, and sewage and hygiene is a big problem. There's a lack of clean water. This is why I think we're seeing the epicentres of bacterial resistance coming out of centres like Asia and other parts of Africa, and obviously coming through to here. And doubtless things are circulating back again. And we still need antibiotics because we know if we don't get antibiotics in time, you don't do very well if you have sepsis. But what's in the pipeline? So clever things, okay? They're combining the old-fashioned cephalosporins with other combinations like clavulinic acid and co-amoxiclav. They're putting them together, and these are working, so that's great. We're using much higher doses of what we thought were toxic antibiotics to get through these problems. We're picking up in the lab quicker. And the FDA in America is trying to push through antibiotics much quicker on the antibiotic pipeline with the pharmacy industry, but it still takes 10 years to get these through. Antimicrobial peptides, there were some amusing articles about frogs a few months ago, where if you can lick the skin of a certain frog, you get some antimicrobial properties, but actually that's true, these are naturally occurring peptides. We're just understanding more about this now. And I know amazing work's been done in the Amazon rainforest looking for antimicrobial peptides. There's huge effort. Every hospital is trying to do what's called antibiotic stewardship. So we try and use what we have left wisely. We have antibiotic apps on our phones, we've got it on the internet, very tight controls on drug industry interaction now with doctors, so they can't push favourite antibiotics. We use a very much mosaic approach here. We get data off on our resistance, and we adjust our antibiotic policies accordingly. And we educate, and also we're educated back, because we're not perfect. And prevention of transmission, this is really fundamental. It's about getting things simple. So we're very lucky here in the UK. We have a Health Act, everything is in UK law. Where I was working before in Singapore, this was not in law, there were no health acts, so we had nothing to sort of go against. So what's brilliant now is we are all accountable. We work to targets sent by our commissioners, and you can see, and I can see, your local hospital data, which is fantastic. If I said this to my Singapore colleagues, they felt very worried and threatened by this. I think this is a fantastic idea. Open and transparent data is key. And hand hygiene is critical here. This is a picture of a hero of mine, Mr. Semmelweis, who was an obstetric gynecology doctor in the 17th century, who showed that if you washed your hands when the babies and the women were being delivered of children, the mortality rate went down by 20%. Unfortunately, he wasn't very popular because at the time, you had to use chlorinated lime for your hands. Of course, it was quite destructive, so his colleagues weren't very happy with this, and he ended up dying age 47 in an asylum. But he was the father of infection prevention and control, really. And if you look around us, and around us now, you think, can't see any bugs, there's no problem. But they've been very clearly shown that naughty bugs like enterococci, Acinetobacter, stick to pens, they stick to papers, they stick to my lanyard. So it's an uphill battle. So hand washing is fundamental to everything that we do. And with WHO is quite clear on this, and we try and have a bare below the elbow approach here. And 30 to 40%, if you look at the literature, of all hospital-acquired infections are attributed to cross-transmission, either by hand hygiene or by shared equipment. So we're very keen here, we don't do any, if we have shared equipment, we're cleaning, we're very tough on hand hygiene. And this is a certain colleague of mine at Winchester, wearing a lovely expensive watch. And if you see, we've actually done some bacterial swabs of his hands, before and wrist area, with washing both with the watch on and with not, and there's very few bugs. There are actually a few in the one where he's doing bare below the elbow, but at least you're reducing the possibility of transmission of pathogenic or non-pathogenic organisms. The other way of looking at this is, yes, we all carry E. coli, but certain E. coli are more pathogenic than others, and so Glaxo is focusing on a vaccine trial as we speak, looking at vaccinating against a certain type of uropathogenic E. coli that only really affects the transitional epithelium of the bladder. So this is very interesting work, and it could be something for the future. And I hear you draw your breath in now. Could we be looking at fecal transplants for people who are colonised with these resistant bacteria? Because it looks as though if you're colonised, you'll be colonised for years and years and years. Well, this is possible. We use it for Clostridium difficile, for people who've had recurrent infections. And the idea is you're trying to repopulate your slightly damaged gut flora with somebody else's bacteria. And obviously it's all screened. And we've done 2 fecal transplants in the trust so far, quite successfully, but not for this reason. But I think this is a future area. Very interesting. The Russians are really keen on this, and the Polish and French have been quite keen on this. Every single bacteria has a virus that will kill it, called a bacteriophage. So there's a lot of research going on to see whether these could be useful. Obviously, delivery of these is going to be tricky. You can't give it into the blood. It would have to be the fecal oral route again. And this is fundamental. And I was at a meeting in the spring with some big thinkers in this area, and I said, we need a collaboration here. How do we stop colistin being used in the world? Can we speak to, say, the Chinese about trying to stop its use in poultry farming? And this is fundamental. And I think there are moves afoot, but this is going to be very, very, very difficult. And try and track and block sales of fake drugs. These are, we're not even talking about gram-negative, about malaria drugs now. And really we need to improve the hygiene and water supplies in the countries where these bugs are coming from. But you are chasing your tail a bit, really. And just to reassure, we've got very, very few nasty bugs here. Okay, we track them. What we do, we monitor resistance, we do antibiotic policy reviews, we screen, if people have come from a high-risk hospital or have come from abroad, they're quietly popped into the side room, we'll screen for these. And infection control, I want to thank the board and Mary for this, is an absolute priority for the trust. So if we get the fundamentals right, we can keep all of these at bay. We've got a very active infection microbiology team, and we work closely with our local GPs and hospital staff. An amazing